Hi everyone! In today's lesson, we will cover research summary passages on the ACT. This lesson is the third and last in our series on the ACT science passages.
Pay close attention to how this passage type differs from data interpretation passages, especially, but also conflicting viewpoint passages. Important questions to ask yourself are, how do the tables and graphs for this passage type compare to those in data interpretation passages? How do the setup and execution of each experiment compare to each other? And what claim do the results of each experiment seem to further? If you're struggling answering these questions or with anything on your assignments, consider an upgrade to test coaching. Let's take a closer look. Okay, before we get started with our first problem this week, let's go ahead and take a look at the structure of this type of passage. They're asking about experiment 1, so I've only included experiment 1 from the entire passage and also the introduction. Well, they're telling you here that sometimes a color change is going to occur when substances are mixed, and that factors such as temperature and concentration, as well as pH, are going to affect how the color change occurs. They tell you a couple more things about what the experiment will be, then they describe the experiment, and then finally, they give us a table with results. Now, in data interpretation types of passages, you could have gone directly. They're asking about something specific to a table, they're talking about the results, and try to match up the table with the answer choices. But our research summary passages are usually going to require a much closer read of the setup of the experiment. Now, you'll be able to recognize research summary passages because they'll usually be divided into different sections, all of which describe different experiments or trials or something of the sort with different results that are going to inform one overall central question. Whereas if you remember from last week, the data interpretation passages were going to instead be focusing on there's only going to be one experiment being described for the entire passage. And for the conflicting viewpoint passages, which we also looked at, there's going to be very little data and different opinions on what that data means from two different scientists. So instead of saying experiment 1, experiment 2, experiment 3, there's usually 3, it'll say scientist 1 and scientist 2. So completely different setup here. Okay, so we know that this is going to be a research summary, so we need to read very closely. What we see here is that we're told that we're going to use potato juice extract, and that the original 3 solutions of procatechol, which are originally colorless, were kept at different temperatures, 0 degrees, 25 degrees, and 100 degrees. This you could have figured out. The fact that it was originally colorless, maybe not. The fact that it was from potato, maybe not. That sort of thing. And it may have something to do with how we're going to answer the question. So you want to read closely. 3 drops of potato juice were then added to each procatechol solution. So potato juice was added to the colorless procatechol. And then the subsequent changes in color were recorded after 5 minutes, and then again after 30 minutes, which we see here in table 1. All right, so now we have a much better idea of what the table is representing than we could have been able to guess just from looking at the table alone. This is going to be the case for every research summary problem that you see. You can't just look at the tables and the graphs and sort of guess what's going on after reading the introduction, which was possible with the data interpretation passages. Alright, all that to say here. So how would we solve this? At which temperatures does the color of the procatechol and potato juice solution change from clear to a shade of yellow? 0 degrees only. Well, at 0 degrees, we know that they all started out colorless. After 5 minutes, we've got yellow, faint yellow, and after 30 minutes, just yellow. So that makes sense. 0 degrees looks like the right answer. Let's see if we found something better. We're going to cross something out. 25 degrees only. Well, 25. Remember, the original was colorless, and then after 5 minutes, we again got yellow. After 30 minutes, bright yellow, but still yellow. So we went from colorless to yellow also at 25. So since 2 of these are correct, we can cross both of them out, as we saw a little bit earlier with our answer elimination exercise. All right, well, both 0 Celsius and 25 Celsius looks good. 100 degrees Celsius only won't make sense if we know 0 and 25 work, but let's see why it doesn't. At 100, we go from originally colorless, but then to colorless, and then after 30 minutes, colorless again. So that allows us to just not pay attention to that last bit there. And that makes answer choice C the correct answer. Now, if you had not done the work here of recognizing this is a research summary passage and reading then the description of the passage very closely before looking at the table and the results, you have to understand the setup of the experiment before you look at the results of the experiment. You wouldn't have been able to reach a conclusion about this. Very likely, you would have thought, well, in no instance am I given here for a transition from colorless to yellow. The transitions here seem to be from faint yellow to yellow. So you need to know that something that is not provided in the table is being taken into consideration. This next question, much like the last one we saw, is going to deal with one experiment alone. So I've only included experiment 2 for this example. Now, they are telling us that we're going to continue using all Procatocol solutions. They told you in experiment 1, and you need to remember that these are originally clear. That's something that we saw there. But other than that, it seems to be the same as before. So varying levels of phenylthiourea now and potato juice were then added to the Procatocol to each test tube. The quantities of phenylthiourea and potato juice, as well as the resulting color change of each test tube, is recorded in the presented table below. Once again, they're not giving you information in the table that's important for the setup of this experiment. The fact that the Procatocol is what we are adding phenylthiourea and potato juice to. All right. So according to the results then of this experiment, which of the following statements is most supported after 5 minutes of observation? So we can get rid of this 30-minute part here. That's not what we're looking at. Only this here. Okay. 10 drops or more of phenylthiourea alone could result in a bright yellow color. Okay, so we would need 10 or more. So we are only looking at this here, and this here. But then we would need, since it says alone, we would need 0 drops of potato juice to also be added to the Procatocol. But we have absolutely no example here in the potato juice column of 0 drops. So we can't really tell. Maybe, but then that wouldn't be most supported. So that likely crosses this out as the answer choice. Answer choice B says 3 drops or more of potato juice could result in a bright yellow color. Potentially, so 3 drops seems to result in a bright yellow color. It's true, potentially more. It could result. So yeah, at least it could, as we see here in the first example. However, it seems to beg the question here, well, could it have been the potato juice alone, or is it the mixture of the potato juice with the phenylthiourea that when they're added to the Procatocol actually produces a bright yellow color? Could be. Maybe we'll find something better. A minimum of 20 drops of phenylthiourea and 5 drops of potato juice are necessary to produce a bright yellow color. Okay, well, we can test that. Answer choice, the first example here has 20 drops. We've got a minimum of 20 drops. But then a minimum of 5 drops of potato juice would also be necessary. But we have three drops. And after 5 minutes, which is what we're looking at, we still have bright yellow. So 5 is not the minimum that we're going to be looking for. It's not necessary to produce a bright yellow color. It can, but it's not necessary. So that can be crossed out immediately. So it looks like the answer choice B is the answer so far. Let's check this one out. A minimum of 20 drops of phenylthiourea and 3 drops of potato juice are necessary to produce a bright yellow color. Oh, well, there we go. So that satisfies this result. Let's see if anything else belies that. So we've got 10s here. Maybe if any result in a bright yellow, we would know. We have bright yellow here, and those are only 2 examples after 5 minutes. So 20 and 5 produces it, and 20 at 3 produce it. We have no other way to test this. So since 3 is less than 5, it would seem to be that what's most supported is that 3 drops of potato juice and 20 drops of phenylthiourea do produce bright yellow. So this seems like the right answer after all. This one is a bit vague. It doesn't include that the potato juice also needs a phenylthiourea to produce a bright yellow color when added to the Procatocol. So we can get rid of that answer choice as well.
In this question, we're asked to look at experiment 3. And we're given a new pH value of 8. And based on the experiment trends in table 3, we're asked to predict what the color of the paracatechol would be at a pH of 8. So In the experiment, the pH values of 4, 7, and 10 are listed, and the procedure, which you don't really need to know, it's the same as the procedure described in the question, is that 3 drops of potato juice are added at room temperature, and then the question we're asking is, what is the color after 5 minutes? So if we look here in the table, we have 7, and we also have 10. 8 is between 7 and 10. So we can actually expect a color between this yellow and colorless because 8 is going to be between 7 and 10. So if we imagine a color that's between yellow and colorless, it's probably going to be a yellow color, but less yellow. So if we look at the answer choices, let's see if we find anything similar to what we would be expecting. Well, dark yellow seems like it would be more yellow, so we can rule that out. Yellow is the color at 7, but we're moving towards colorlessness, so we can cross out yellow. We can cross out colorless because it doesn't become colorless until 10. So our best guess here, although we don't know for sure, but we don't need to know for sure, is that we can expect a faint yellow. So that's going to be our final answer.
Okay, so let's start by reading the question here. To better understand the implications of the data in experiment 2, which of the following conditions should be identified and kept constant for each test tube? Okay, so I don't really know what experiment 2 is about without double checking it down here. So I'm going to just take a look and get a sense of what it's about here. A series of tubes were set up, all containing pyrocatechol solutions of the same concentration. Varying levels of phenylthoria and potato juice were then added to each test tube. The quantities of phenylthoria and potato juice, as well as the resulting color change of each test tube, is recorded and presented in Table 2 below. Okay, so I'm looking for what's going to be kept constant, what needs to be kept constant for this to be good experimental design, which means what I'm going to look for is what's changing. I'm going to look for what's changing in the table or what changes are being recorded and try to figure out what the constant needs to be in order to look at those changing variables. So it looks like we're seeing varying, so that's a word that I think is probably pretty important when I'm thinking about constants and change, varying levels of phenylthoria and potato juice. Okay, so if those are varying, and then it says up here that the pyrocatechol solutions are of the same concentration, it seems like we've got these two things that are changing and one thing that's staying the same. I'm just going to take a look at our table and see. So it looks like we have different amounts of phenylthoria, different amounts of potato juice, and then different effects from those things. And the pyrocatechol is the same concentration. So let me look at my answer choices here. It looks like they all have to do with pyrocatechol. So let me read these one by one. The original concentration of each pyrocatechol solution should be kept constant. Well, it says that right here in the experiment. It says they're all containing pyrocatechol solutions of the same concentration. So my first instinct is that that's probably going to be right, but I'm going to take a look at the rest of my answer choices just in case. The temperature of each pyrocatechol solution, we don't necessarily have information that tells us it needs to be kept the same in the experiment, but we do know from experiment 1 that using different temperatures of pyrocatechol can actually have an effect on color, so it seems like we might want to keep it the same. The original pH of each pyrocatechol solution, well, same thing. If we look over here at experiment 3, we can see that the pH changing has an effect on color. So that might need to stay the same. And here we go. Last answer choice says all of the above should be identified and kept constant. Well, we know that at least one of them needs to be kept constant, and the other two should probably be kept constant. Given good experimental design, you never would change more than one variable at a time, it seems like the last answer is just pretty obviously true at this point.
All right, now this question is a bit more tricky than the other ones we've looked at so far. So they're saying, suppose in experiment 2, there was an error in making the phenolketonuria solutions added to each test tube, and that the second half of the test tubes received phenolketonuria solutions with double the concentration of those as the first. So the idea here is the first and the second will should have the same amount of solution, and this is what the original example did. But now in this mistake example, in the one that actually applies to this question, the original will have the same as it would have, or I guess one of them will have the same as amount that they had in the original, but another one will have twice that amount. Okay, so how would the number of drops of phenolketonuria required to produce the same results need to change? Okay, well, it may be that since we made a mistake here and we have double the amount of the phenolketonuria in the second beaker, maybe we just need to remove half of it, or maybe we just need to increase this one by 2. But maybe all they're saying is we want it to produce the same results. Maybe we don't actually need to do that. So let's go ahead and check out our answer choices to see whether or not these changes produce the exact same results after 5 minutes and 30 minutes for all of the considerations in this table. So answer choice A says the first set of test tubes receiving the original concentration of phenolketonuria would need half the number of drops of phenolketonuria as those receiving the double concentrated solution. So this one now, in the mistake, it would actually be, or the way we would fix it for answer choice A would be to say, well, half of our test tubes are going to have half of the original amount, and half of them here would have double the original amount as per the mistake that was originally described here. Well, the thing is, when we look at our table, we're not considering any change in the potato juice at all. We're simply told that half of the beakers of the phenolketonuria are going to see this change and half of them will not. How do we know which halves? And how do we know when those halves paired with potato juice, what the result, when those are actually paired, what the results are going to be after 5 minutes and 30 minutes? If we look at maybe some constants here, when the potato juice remains the same at 3 drops, and we half it from we half the amount of phenolketonuria from 20 drops to 10 drops, the color becomes more faint. Then by definition, when we increase the amount of phenolketonuria, the color becomes more bright if we double it. So it looks like we're going to get a different result by doubling or halving the amount of phenolketonuria. So that doesn't make sense. We can get rid of answer choice A that way. The first set of test tubes receiving the original concentration of phenolketonuria would need twice the number of drops of phenolketonuria as those receiving the double concentrated solution. Okay, well, if what we do here is we also double the one that has the original amount, so we make both of these have some extra phenolketonuria, what would happen? Well, if adding more is going to make things more yellow, what will happen here very likely, if we have the same amount of potato juice, is the now 40 drop phenolketonuria beaker will have an even brighter shade of yellow. So brighter shade of yellow. And the 10, that would now be 20 drops, would just have yellow, or potentially bright yellow. But what exactly is bright and what exactly is faint? If everything is brighter by about the same amount, by comparison, the faint yellow, or the faint yellow, which would be less faint than originally, would still be significantly more faint than the now brighter bright yellow. So by comparison, the 20 drop versus the 40 drop would look like a faint yellow. Bright and faint are what we call subjective descriptions. They're not objective. They're not really telling us anything numerical that we can actually test. So by comparison, we could say, well, the yellow will remain fainter for the ones that have fewer drops if both are the same. So this looks really good. Let's see if we find something a little bit better. No change. The first set of test tubes receiving the original concentration of phenolketonuria should receive the same number of drops of phenolketonuria as those receiving the double concentrated solution. Well, we're talking here about concentration and drops. So it will be the same as when here in the example where we have 20 drops of something and 10 drops of something that have the same concentration, right? We have a difference here of bright and faint. Well, if we add more drops of something with fainter concentration, that's not necessarily going to be the same as adding half that amount of a greater concentration. We're just not very sure here. We don't have enough data to know specifically, how much phenolphthalein there is per drop, what percentage of the extra concentration will go there. We're not very sure about any of this. So this looks very, very difficult to find out for sure. We don't know. And the first set of tubes receiving the original concentration of phenolphthalein should receive the same number of drops of phenolphthalein as those receiving the double concentrated solution. However, the number, so the same as we were saying before, okay, so a little bit convoluted, the number of drops of potato juice added to the first set of test tubes should double. Okay, well, if we now double the number of the amount of potato juice, what would happen? We didn't have any examples where we double the amount of potato juice, but we see something similar when we go from 1 drop of potato juice and 10 drops of phenolphthalein and 3 drops of potato juice and 10 drops of phenolphthalein. I'm looking at these two because the only thing that's changing here is the potato juice. And it looks like after 5 minutes, the results are about the same, and after 30 minutes, the results are about the same. So if in these two, the potato juice is the only change, and that's not going to change the results, then these two answers are basically saying the same thing. So just a second ago, I was wondering, I'm not sure, it seems complicated, how do I know? Well, if two of our answer choices are virtually saying the exact same thing, they can't both be right. So we can go ahead and confidently now cross both of those out. And that leaves answer choice B as a correct answer.
Okay, let's start again by reading our question. Assuming that in experiment 2, the test tubes were kept at 25 degrees Celsius, which of the following observations seems to suggest an inconsistency between the results of experiment 1 and the results of experiment 2? Okay, so that means that we're going to need to look at both of our experiments here and have some understanding of what's going on. So the first thing that I see is that we're saying that in experiment, we're adding a condition here. So we're adding something that's not written in the description of experiment 2. We're saying that the temperature here was kept at 25 degrees Celsius for this whole experiment. So that's our premise. Which of the following observations seems to suggest an inconsistency, so something that seems to be awry, between the results of experiment 1 and the results of experiment 2? So I'm going to go back up to experiment 1, and I'm going to hone right in on seeing that in experiment 1, that was the experiment where the thing we were changing was temperature. And in particular, I have a row here that shows us the result of a solution temperature of 25 degrees Celsius. Okay, so I have a feeling that that row is going to be important, that row of data. And at this point, now I'm probably going to need to read through my answer choices because I'm going to need a little more information about what I'm looking for. Okay, so let's look at the first one. In experiment 2, when 3 drops of potato juice and 0 drops of phenolphthalein are added to the pyrocatechol solution, the resulting solution is colorless. Let's just double check to make sure that that's true to begin with. 3 drops of potato juice, 0 drops of phenolphthalein added to the pyrocatechol solution, the resulting solution is colorless. So I'm going to go up to table 2 here, and I'm going to look for 3 drops of potato juice and 0 drops of phenolphthalein as described. And yes, indeed, it is. The resulting color is colorless. Okay, so that part at least is true. We would expect the same results from the experiment held at 25 degrees Celsius in experiment 1. Okay, so it's basically saying, well, it's colorless here, it's yellow and bright yellow here, and we would expect those to be the same. I'm not sure if we'd expect them to be the same because I don't know if the 3 drops of potato juice and 0 drops of phenolphthalein is what was added in experiment 1. So let me double check that. It says that we have solutions of pyrocatechol kept at 25 degrees Celsius We added 3 drops of potato juice. Okay, so that part's right. And I don't see in here anything about phenylthuria, so we can assume it was 0 drops. They would tell us if they had added it. So I would say yes, we do. We would expect these to be the same result because you have the same temperature and you're adding the same thing to the same solution. So it should be the same result because we got 5 minutes, 30 minutes, 5 minutes, 30 minutes. Okay, so now I could select this and say it's the right answer, but what I see is that I actually have one option. If I look ahead, right here, option 3 says both A and B seem to suggest an inconsistency. So I have to actually rule out my second answer choice. I can already rule out my fourth answer choice where it says neither of them's right because I know that the first one's right. But I need to rule out the second choice in order to choose an answer. In experiment 1, after 30 minutes, 3 drops of potato juice resulted in a bright yellow color in the solution. All right, that's true. That's the thing that we just checked. We would expect the same results from every test tube where 3 drops of potato juice were added in experiment 2. Now this sounds basically like the same thing as we just read in option A. The difference is it says we would expect the same results from every test tube where 3 drops of potato juice were added. Well, let's see. Every test tube where 3 drops of potato juice were added would be this one, and this one, because there's 3, 3, 3. The difference is that we have 10 drops of phenylthuria, 1 drop, and 0 drops. Well, we wouldn't actually expect those all to be the same because we have a changing variable. We have a changing amount of phenylthuria, and that is not present in experiment 1. So we can rule out answer B as an expected, that's not an inconsistency. That seems totally reasonable. So we can rule out that one. By virtue of ruling that one out, we can also rule this one out, and we are left with a correct answer of A, which was our initial instinct.
I hope you found this lesson to be helpful. We took a look at the structure of the research summaries passage and how that structure can be of help in answering its associated questions.
We saw that each sample question required understanding multiple trials of the experiment at once, and therefore much more targeted reading than other sections, such as data interpretation or conflicting viewpoints. When you do your research summary problems this week, keep in mind that the strategies we discussed apply to other passages in this category.
Happy prepping!